Welcome to our video about the fascinating world of Pokemon and their placement on the Tree of Life. Today, we take a closer look at Starmie and its connection to all Deuterostome Pokemon. Ready? Let's go! The Deuterostomes represent a superphylum within the Bilateria, bilaterally symmetrical animals, from which the chordates, chordata, including humans, and the echinoderms, echinodermata, are the primary taxa. One of the two defining characteristics of the Deuterostomia is the further development of the blastopore during embryogenesis. During gastrulation, the blastopore becomes the anus, and the mouth forms anew. This process is known as deuterostomy. The second distinguishing feature is the dorsal backside positioning of the central nervous system, CNS. In contrast, among the other bilaterians, the protostomes, the blastopore develops into the mouth and the anus breaks through secondarily. The CNS is located ventrally, on the belly side. Developmental biology began in the 19th century. Early embryonic stages were closely observed and described under the microscope. The contributions of the Baltic German naturalist Karl Ernst von Baer and the German zoologist Ernst Haeckel proved to be particularly pioneering and influential. Scientists discovered that the formation of the digestive tract in triploblastic animals occurs in two distinct ways. In one case, the blastopore becomes the future mouth. The developing digestive tract grows through the embryo, and the anus breaks through at the end. In the other case, the blastopore becomes the future anus. The digestive tract similarly grows through the embryo, and the mouth breaks through subsequently. This developmental biological distinction was first interpreted in evolutionary terms in 1875 by the British biologist Thomas Henry Huxley, who published his findings in two separate works. Huxley regarded the mode of digestive tract formation as a trait that had evolved in very distant ancestors and had since been inherited by their increasingly diversified descendants. Accordingly, all living triploblastic animals today descend either from an ancestor whose embryonic blastopore developed into the mouth. These are the protostomes, or from an ancestor whose blastopore became the anus. These are the deuterostomes. Thomas Henry Huxley named the first group the protostomes, Archaeostomata, and the second group the deuterostomes, Deuterostomata. Both terms were introduced to a broader audience the following year by the Irish anatomist Alexander McAllister in his textbook. However, the concepts of Archaeostomata and Deuterostomata remained confined to the English-speaking scientific community. As a result, 12 years later, the Austrian zoologist Berthold Hatschek coined a new term, Zygonera, to describe the animal group that Huxley had previously referred to as Archaeostomata. However, Hatschek did not offer a corresponding synonym for Huxley's Deuterostomata. This terminological gap was only closed in 1908 by the Austrian zoologist Karl Graben. In his treatise, The Systematic Classification of the Animal Kingdom, Graben initially adopted Hatschek's term Zygonera, but later introduced his own synonym, Protostomia. He contrasted the Protostomia with the group of triploblastic animals whose blastopore becomes the anus. This group he now called Deuterostomia. 33 years after Huxley's Deuterostomata, Graben had coined a nearly identical term for the same concept. Throughout the 20th century, Graben's terminology became widely accepted in developmental and evolutionary biology. Hatschek's Zygonera, as well as Huxley's Archaeostomata and Deuterostomata, gradually fell into obscurity. Today, the biological sciences typically use only the terms protostomia and deuterostomia. Following the discovery of deuterostomy, this developmental trait helped unite the seemingly disparate animal groups of the chordates, chordata, hemichordata, and echinoderms, echinodermata, into a single lineage of descent. This lineage was given the name Deuterostomes, literally second mouth animals or new mouth animals. However, embryological studies revealed that deuterostomy-like development also occurs in a handful of other groups, including arrowworms, Caedognatha, some Lophophorids, Lophophorata, certain horsehair worms, Nematomorpha, and even some arthropods, Arthropoda. For decades, scientists debated whether these organisms should be included within the Deuterostomia. Thanks to modern molecular phylogenetics, phylogenomics, the answer is now clear. Chordates, hemichordates, and echinoderms do indeed form a true monophyletic lineage, while the other groups represent cases of convergent evolution. independently arriving at similar traits. Within bilaterians, bilateria, the Deuterostomes belong to a larger grouping known as Nephrozoa. Their sister group is the Protostomia, meaning that the last common ancestor of both groups was itself a bilaterally symmetrical nephrozoan. These early ancestors already showed directional movement with a defined head end, where nerve and sensory cells concentrated, a process known as cephalization. They also developed 3 germ layers and excretory organs known as nephridia. Molecular clock studies suggest that these nephrozoans lived in the late Proterozoic, likely around the middle of the Sturtian glaciation during the Cryogenian period, about 680 million years ago, before the lineages of Deuterostomes and Protostomes finally diverged. Roughly 30 million years later, the Deuterostomes themselves split into 2 major branches still alive today: the Ambulacraria, which include echinoderms and hemichordates, and the chordates. This key divergence occurred during the interglacial period between the Sturtian and Marinoan ice ages. The Cryogenian was followed by the Ediacaran period, when further branching took place. Already by this time, echinoderms, hemichordates, lanceolates, leptocardia, Christozoans, and tunicata, had begun to evolve independently, well before the Cambrian explosion. These earliest Deuterostomes were soft-bodied, only millimeters in size, and have left little trace in the fossil record. It's only in the early Cambrian that we begin to find recognizable Deuterostome fossils, still small, usually only a few centimeters long. One exception was the Vetulicolia, an enigmatic branch of Deuterostomes, with species like Vetulicola longbowshanensis reaching nearly 10 centimeters in length, huge by comparison. The last common ancestor of all Deuterostomes likely had a worm-like body plan. Whether it was sessile on the seafloor, benthic sessile, actively crawling, benthic vagile, burrowing through sediments, subbenthic vagila, or swimming freely in the water column, pelagic vagile, is still uncertain, though the swimming lifestyle is currently considered less likely. The Ambulacraria retained mostly benthic lifestyles. Early echinoderms, for instance, were still bilaterally symmetrical and crawled or hopped across the seafloor. But by the early Cambrian, many echinoderms had abandoned bilateral symmetry in favor of asymmetry or radial symmetry. This dramatic evolutionary shift was accompanied by a move from active bottom dwelling to more sedentary, sessile lifestyles attached to the seafloor. Permanently sessile animals no longer require a body plan oriented toward a single direction of movement. All modern echinoderms descend from such sessile pentaradial ancestors, possibly originating from the fossil genus Camptostroma. Only their larvae retained bilateral symmetry. Apart from the still sessile coeles, other echinoderm groups later returned to benthic vagal lifestyles. This includes the closely related feather stars, as well as all Eleutherozoa, sea cucumbers, Holothuriidea, sea urchins, Echinoidea, sea stars, Asteroidea, and brittle stars, Ophiuroidea. Interestingly, nearly half of all modern sea urchin species, the Irregularia, display signs of secondarily evolved bilateral symmetry, a trait that is fully developed in sea cucumbers. The other branch of the Ambulacraria, the hemichordata, also adopted benthic movement. Acorn worms, Enteropnusta, burrow through the seafloor, while pterobranchia, live in sessile tubes, although members of the Cephalodicidae family move freely across the seabed without ever constructing tubes. Chordata, however, took a very different path. They evolved into free swimmers. Their elongated, laterally flattened bodies were stabilized by a dorsal notochord. Modern lanceolates, Leptocardia, rarely swim actively, instead burying themselves tail first in coarse sand or lying flat on firmer seabeds. The earliest olfactors likely led pelagic lives. Early Christozoans followed this model, as shown by fossils of Miloconmingiida, Picaeidae, and Conodontophora. Meanwhile, tunicata, adopted a benthic sessile lifestyle as early as the Lower Cambrian, though they retained pelagic larval stages. Adult tunicates lost bilateral symmetry, but later, groups like salps, Thaliacea, and appendicularians, copulata, returned to pelagic lifestyles, with appendicularians keeping their bilaterally symmetrical larval form throughout life. The extinct Vetulocystida and Vetulicolia are known only from Cambrian fossils. Vetulocystida were bilaterally symmetrical, with 2-part bodies consisting of broad anterior sections and tails. they likely lived mostly anchored in sediment, occasionally moving by flicking their tails. Vetulicolia shared a similar two-part body plan, but with laterally flattened anterior regions and tails attached near the dorsal posterior end. They were probably sedentary seafloor dwellers, but capable of swimming when threatened. Some genera diverged. Banffia became burrowers, while Vetulicola developed into active swimmers with streamlined bodies. Among modern echinoderms, the order Forcipulatida, mostly large sea stars, comprises 6 families. They feature a mesh-like pattern of calcareous plates, lack marginal plates and paxillae, and possess straight and crossed pedicelaria on stalks across the body. Their arms are flexible, rounded in cross-section, and lined with ambulacral tube feet, typically arranged in 4 rows, equipped with powerful suction discs. Most Forcipulatids have 5 arms, but species in the families Heliasteridae and Pycnopodidae often possess many more, giving them a superficial resemblance to the unrelated sun stars, Solasteridae. Forcipulatids are predators and scavengers capable of averting their stomachs for external digestion. The family Asteridae, with nearly 170 species across 40 genera, is the 3rd largest sea star family after Goniasteridae and Astropectinidae. It includes most sea stars found along European coasts. Members of Asteridae have a mesh of calcareous plates in their skin and longitudinal rows of simple spines. Both straight and crossed pedicellariae are typically present, and an anus is always present. Like other sea star families, they can avert their stomachs for external digestion. However, Asteridae are uniquely capable of forcibly opening bivalve shells, brachiopod valves, or snail opercula using the strength of their tube feet and arms, even cracking sea urchin tests, as seen in the ice star, Marthasterias glacialis. Not all sea stars with strong tube feet can access prey this way. Most species in this family inhabit temperate and cold waters, though some occur in warmer regions. The common sea star, Asterias rubens, is widespread throughout the Atlantic, including the North Sea and Baltic Sea. The ice star, Marthasterias glacialis, is also found in the Mediterranean. This species and the sunflower sea star, Pycnopodia helianthoides, from the North American Pacific coast, whose many arms resemble those of sun stars, are the largest known sea star species, reaching diameters of up to 1 meter. While many species prefer slow-moving prey like mussels, snails, barnacles, or sea urchins, Stylisterias foreri and Astrometis certulifera can capture decapods and small fish using their pedicellariae. A keystone species in the North American Pacific ecosystem is Pisaster ocraceus, which feeds on numerous animal species and is the main predator of the otherwise dominant California mussel, Mytilus californianus. In Australia, the invasive North Pacific sea star, Asterias amurensis, has become a threat to native species. That's it for now. In the next episode, we take a closer look at Mr. Mime and its surprising connection to Hitmonlee. Feel free to subscribe to our channel, leave a like, and check in next time.